Okay, thanks for coming. Today it's my pleasure to introduce Jordon Beckler, who's going to talk to us. Make sure my notes are right. So you did your BS and your PhD at Georgia Institute of Technology.
Yes, sir.
Did his PhD in 2014. A few other positions. He was at Moat for a while. Now he's an assistant research professor, I think, at FAU Harbor Branch. And he's going to talk to us today about blue holes, but also has a lot of interest in autonomous sensors for nutrients and also iron and sediments, which is what I'm interested in.
Thank you, Tim, for hosting and for the invite. It was nice to meet a lot of old faces, but then a lot of new faces as well. Good group of students today at lunch. That was a lot of fun. So thanks again. Okay, let me get my bearings here. I have one of these, presumably. Yep. So I'm going to talk about two different blue holes that are right in our backyard that prior to me starting this project, I had no idea they existed. So I don't know if raise your hand if you didn't know that these things existed in our coast. Still a few. Wow. Cool. All right, so I want to thank, this is kind of my co-author and acknowledgements on Kill Two Birds, but my student Matt, he moved on to wow, brain freeze. USC, my God. Okay, so Matt moved on there. He was probably the biggest contributor to this work. He was involved with most of the cruises. And then my other students and techs, everyone at least participated in once. This whole project was kind of bootstrapped. You know, NOAA, OER, you don't tend to get huge grants from them. And when you do, you got to really make it count. So this whole project was extremely underfunded overall. Georgia Tech, Martial Taillefer, he provided the benthic lander, and then we modified it and deployed it in blue holes. Don Nuzio at AIS designed our electronics for our in-situ instrumentation. The Moat folks, especially Emily Hall, they were really, amazing in terms of getting the project planned out, logistics, doing a lot of the ecology stuff. Chris Smith, right here in St. Pete. Chris was in charge of the radium isotopes. And I don't know why I put Frank Stewart's name down there. I must have cut and paste, but he's also only at Georgia Tech, so pardon that mistake. And then finally, an awesome group of technical divers in even the PBS team from South Florida. So they were pretty epic, and I'll talk about them a bit more going forward, all the logistical challenges involved in this project. Again, it was funded by OER, and then again, bootstrapped with other various funding sources. First, I want to talk just a little bit about my lab and some of my other projects, really briefly, just in case you're interested. I'm starting a dialogue. This is my current lab, great group of students right now. We've done work, a lot of work in this region. We really tend to focus on the Lake Okeechobee and its conveyances, and then the effects, for example, with algae blooms across the way. And now we're down in Florida Bay quite a bit, working as well because it's much prettier and not as disgusting as Lake Okeechobee. So my research interests are primarily, it's hard to, I'm sure as many of us have this problem, it's hard to kind of gather your research interests into a specific box, but redox processes at environmental interfaces, carbon, iron, sulfur, your typical culprits, with redox cycling, and then of course coupling to nutrient cycles. Sediments in particular and their effects, their ecosystem effects like eutrophication, algae blooms, and hypoxia. We develop in situ sensors and platforms, primarily platforms, to actually go and make measurements autonomously at a higher spatial and temporal scale. I called this one biogeochemical exploration. It's kind of a term that I made up, but this blue hole project didn't really fit in any other box. And last but not least, something I'm really actually passionate about is the concept of sediment health. Just as we appreciate soils and air and water, you never really think about what is healthy sediments, right, from a societal sense. Just a little bit on that. I perceive it, I perceive sediments as having various resilience buffers, various degrees of these things, and you get external stressors, either anthropogenic or climate related. And ultimately, you could frame all of these environmental issues to some extent by this kind of trade-off here. And this is related to the blue holes a little bit, but this is a figure how I perceive a nice, healthy, oxygenated system, like the new Tampa Bay, for example, kind of, right, where you have a pretty biodiverse environment, lots of oxygenation. So that's key from my perspective. A lot of oxygen keeps a nice, healthy redox buffer, an iron oxide redox buffer. My dissertation was about iron, so I tend to be, pretty focused on iron, but to a large extent that can help alleviate nitrogen and phosphorus fluxes outwards. They undergo chemical transformation or they can absorb onto these iron oxides. The iron also keeps the hydrogen sulfide at bay as well by forming or via the iron sulfide buffer system. On a perturbed system where you don't really have like a stagnant old Tampa Bay system, you have a lot of organic matter funneling down into the system, which consumes the oxygen so fast that you start accumulating hydrogen sulfide due to anaerobic sulfate respiration. It's really the only electron acceptor left. And you end up producing very high pore water nitrogen and phosphorus concentrations, and then those can diffusively flux out of the across the stem and water interface. So keep that in mind. This is kind of like what the inside of a blue hole is going to appear like, even though it's not necessarily a polluted environment per se. Finally, about the sediment aspect side, so there is an applied component to this. Sediments you may not realize, and I'm sure many of you probably do, but there probably is some stuff you may not have realized, like how sediments can be used to generate energy. And then the obvious ones like restoration as well. So always interested in the applied sciences associated with sediments. And I think this is the last slide, just about other stuff. Some projects that we are currently pursuing or just finished pursuing, and these are the ones that I enjoy talking about. I'm not putting the ones that I don't enjoy talking about. Many of us might have some of those projects, not to be named, but Florida Bay, like I said, we're down there all the time. My student just sent me this data the other day for an EcoHab proposal, but my God, like, come on, man, really, you've been holding on to this massive ammonium flux data that corresponds right to the bloom season when we had a really bad bloom last year. Like, I need to see that, right? So we're looking at Florida Bays, seagrass, hypoxia, and obviously HAB issues. We're working on a NASA project looking at sediment CDOM fluxes to the surface where satellites can see them. A Lake Okeechobee project we recently just finished up, but we're apparently going back out there this next year. We just got this grant funded, which is to deploy. It's a long-term system that we designed for. It's a system for long-term sediment, nutrient, or really anything else that there's an in-situ sensor for. Sediment nutrient fluxes. So it'll kind of be one of the first of its kind, and we're trying to commercialize that through the university as well. So feel free to check that out. All right, on to the good stuff. So again, biogeochemical exploration. Green Banana Spring. This is one of the sites we're going to be looking at, and you can kind of see the progression. Back in the day, it was really just limited to these obscure magazines like Underwater Speleology, where you had these advanced technical divers that were going down there as a hobby. They were going down 400 feet down these caves. Don't try this at home. And you can see in 1 year, they only made it down to this constriction. And then the following year, they managed to get it further mapped. So just kind of providing some background to it. I think the project that we ended up completing was sort of like the first scientific or truly scientific study of the deep systems in the Gulf of Mexico. There's been plenty of work like from Chuan Min, who's worked on the nearshore stuff. This was deep Gulf of Mexico. Please correct me if I'm wrong. Why do we care? Well, there are obviously ecological hotspots. That's why the fishermen and the divers would go down there. They, this is credit from Jim Coulter at Mote. There's several dozen of them that are known. Only a few have actually been explored comprehensively. This is kind of jumping ahead a bit, but it's more, I think it's more than just having the shelter there. I think a lot of what attracts the organisms is the funnel shape of these blue holes, and all of the stuff that just accumulates them gets remineralized down in the mud at the bottom of the blue hole and then fluxes out, sort of, causing a cascade of ecosystem trophic interactions from there on out, laterally. Aquifer connectivity, this one we didn't know, if this was the case, and I won't spoil the surprise, although my abstract might have. It'd be pretty far-fetched to see it 35, 50 miles offshore, right? And there's obviously implications for both outflow into an area like where there's red tide blooms, potentially starting, and as well as inflow via saltwater intrusion even. It's an all marine resource, so we kind of had a feeling that these were going to be stratified environments. The divers have reported their chrome scuba tank stuff turning black from, what we would suspect is due to hydrogen sulfide poisoning. So, and there's several sites around the globe where biogeochemist or geomicrobiologist, I don't know, been up too long today, sorry. They're looking for sites that have this redox stratification, because we can learn about early life on Earth, just about fundamental processes, or life on other planets as well. And finding an all-marine resource, entirely saline environment is actually pretty unique. There's not many of them out there, especially in our region. There's nothing like it in the southeastern US. Of course, paleo work, that's a lot of the other work in blue holes that folks have done across the world, is looking at the last 10,000 years of carbon, atmospheric CO2, that kind of stuff, temperature. And last, I'm no expert on geology here, but how do these blue holes actually form? Let's see if this plays. So yeah, just to get a feel for the environment that's down there. This is from the PBS documentary Changing Seas. So check it out. It's really, really cool. Okay, this is unpublished video from, well, it's on YouTube, so maybe it is published, maybe that counts, but this is from one of the divers who was really, really helpful with this project. This is from Green Banana. You could see all the fishing lines down there, so it is a true death trap. People were getting tangled up. It was extremely sketchy. Hey, look, there's a shark. So yeah, just wanted to show you what that they are indeed ecological hotspots. Perhaps too much so in this case, but all right. Again, not my area of expertise, but we have what we presume is a relict or active spring, right? So just like a spring would form on land, it happened to make its way offshore, or we had sea level eventually rise and cover it. Same idea for the sinkhole variety. Just like on land, you had the collapse of carbonate bedrock, and then 10,000 years ago, when sea level was lower at this point, now sea level has risen, and in our case, it's risen so much that it's actually underwater. So that's my rudimentary understanding of how blue holes form. Now the red tide connection, some of us like Yonggang actually think upwelling is an important source offshore for red tide. Some of us don't necessarily believe that. Sorry, Yonggang. Partially. And when you get closer to shore, you get estuarine outflow. You could see some of the other sources. Blue holes would kind of come in this area if they were indeed important. And the kind of idea, is that indeed some of Bob Weisberg's work from a few years back, he ran tracer studies, forget which conference I saw it at, but he had kind of rewound the hydrodynamics to almost the same area where we find these blue holes, like 40 or 50 miles offshore. So it's possible you get a direct source to the red tide blooms, which we're pretty sure start at depth, and then eventually they can upwell their way near shore. So that's, of course, something that's always in the back of my mind. The two holes we're going to talk about, AJ holes, what I'll talk about first. This is the one that's a sinkhole, a former sinkhole, probably. Green banana, well, AJ, it has this debris pile at the bottom. It's pretty wide at the mouth. I want to say it's probably 200 feet or so in diameter. It's almost a perfect circle. Green banana, much now narrower to the point where it's actually difficult for divers to maneuver our lander package through this hole. So at its narrowest point, it's maybe only 15 feet or so in diameter. And this is the one that we think is a former spring. We know that there's not a huge debris pile down here, not like Amberjack, which is like a massive debris pile, you know, 100 feet tall or something like that. Maybe not that high, but, you know, tens of feet. Activities and logistics. So I'm going to show data from 3 campaigns. We visited each site once in the dry, once in the wet season. I'm only going to show data from the dry season at Amberjack Hole just because the wet season is so similar. You just take my word for it. It's pretty similar. Green banana, on the other hand, I'm going to show both wet and dry season because it is different. Each campaign was about 5 to 7 days, highly coordinated, thanks to Emily at Moat. We had 3 boats, 6 volunteer tech divers or so, you know, plus or minus a few on each campaign, and they can't dive in 2 sequential days just because of the depth, and they only get 15 minutes of bottom time as well, so they're doing a lot in those 15 minutes. There was pre-cruise visits as well. I was involved with these to install some rigging and scuba tanks as well for shuttling because their decompression time is like 3 hours on the way up as well, so they're in the water for a long time. Blue holes are a small target, so it just takes a long time, you know, once we finally get on the site with the science team, 3-point anchors. Visibility, this is a special one because it's so turbid down there, the divers would go down and accidentally pick up the silty sediments, and we could not operate the next day, quite literally, like it's that bad. UPS decided to lose the benthic lander. They lost an entire massive pallet freight, never to be found, so we had to cancel the cruise. And lastly, you know, it's just a lot to shuttle. By the time we finally got back at night, it was late and you have stuff to process. Now, this was, you know, icing on the cake, if you will. It's the nice thing about working with Moat is that there are some fortunate donors. So we actually got to stay in 2 mansions between our team, complete with a private movie theater and 4 swimming pools. So, and it was free. So again, you got to make do with the limited budget. Not so bad. Now this is a busy slide, but I just want to show, you know, this is basically everything we did from the proposal. But I'm going to show data, the geochemistry data. I'm going to show some water column profiles, right, on the scale of about 100 meters or so vertically. And I'm going to show sediment profiles that are more like 20 centimeters or so. The way we did it, we had, well, we did it 2 ways. The divers would literally just lower the lander, which is neutrally buoyant, into place on some cases, but then we decided, you know, kind of depending on the conditions, that lowering it down a line was the more rational approach. Kind of, you know, it also depends on how comfortable the divers are as well. Just want to show sorry, I got to go over here. This is the silt, or actually, no, this is how complex it is. You know, you got to imagine these divers are, you know, geeked up on helium and their mixes. They are, you know, trying to hold lights. They're at 400 feet with limited bottom time, stressing, and they're trying to balance. You know, we loaded these guys up with stuff, and you see them kick up the silt. So some of the Niskins we had to actually collect manually just because you can't lower the CTD package in accurately into the small target. And it did. It got caught a few times on the blue hole wall for green banana, the narrower spring. did I do last time? Just real quick, sediment core is what we, we push the sediments up bit by bit, and then we section the sediments, isolate the pore waters via centrifugation, and then filter them. If any of y'all do this, we got a really cool system. So check with me about it. It's a way to do it anaerobically so you don't sacrifice the redox speciation. This is a cool technique that we used way back, is this 30 years now? My gosh. So what mercury gold voltammetry is, we would use these microelectrodes here. You can see one going into the top of a sediment core. It's a 100 micron diameter gold wire that we amalgamate with elemental mercury. And then we go in small depth increments, so sub-millimeter depth increments, bit by bit. It takes a few hours. And at each depth, we scan a range of potentials, and we measure the current at a counter electrode, a separate electrode. And at certain known potentials, most of the time, we know these potentials, we'll see big, or not necessarily big, but we'll see peaks in current that are proportional to the concentration of said analyte at that potential. And so we can, from that, quantify a lot of different redox analytes, so pretty much everything except the nitrogen system. So we can measure dissolved oxygen, we could measure hydrogen sulfide, elemental sulfur, thiosulfate, so a lot of different sulfur redox intermediates, and then iron chemistry as well, both ferric and ferrous iron, and then even iron sulfide clusters, which are pretty indicative of an active iron sulfur buffering system. So that's just what this profile is. Then we ultimately plot it as a function of depth. Oh yeah, and we also did this on discrete samples in the water column. So we're getting sulfur speciation within the water column as well. The benthic lander, we have, it has a benthic incubation chamber. Again, we neutrally ballasted it. And just real quick, the way this works is you make a little parcel of water in contact with sediments, and then nutrients will accumulate, or oxygen will actually get consumed by the sediments over time. So you'll see an increase in said ammonium over time, and then you just fit the accumulation rate, do a little mass balance, and you can end up with your normal units for sediment fluxes. Now, each deployment only gives you one single flux measurement, so it's a lot of work, deployment, retrieval, and all the setup and logistics just to get one single data point. PBS had a nice, they do a much better job than I can of actually explaining it. There's the divers lowering this thing, neutrally buoyant. A lot of trial and error involved with this. Going into Amberjack Hole. And there they are. They use their scooters sometimes to push it as well. Oh, this is a funny quote. It's a shame we can't listen to it. And eventually the lander ends up on the blue hole floor, hopefully without kicking up too much mud. And then there are syringes, autonomous syringes that are activated in series to actually collect those samples as a function of time. And we analyze them back in the lab later, derive that flux. The last thing, well, one of the last things I guess my group did was we attached an in situ electrochemical analyzer to this benthic lander platform that we deployed for 24 hours or so. And we monitored, we ran the same voltammetry experiments that I showed earlier, but we put 2 electrodes so they would land in the sediments on the benthic chamber, and 2 electrodes just strapped to the lander frame. So we would get, high resolution time points of the water column or sediment redox conditions with respect to oxygen sulfide, thiosulfate. All right, this is the data. So first I'm going to talk about AJ Hole, the water column. And what I'm getting at here is this is a pretty static or temporally static, but extremely vertically dynamic environment here. So just starting with the basic stuff, CT, our salinity, it increases right at the mouth of the blue hole, but then actually decreases as you go down, although your density increases overall. And so it seems like it's more temperature controlled than salinity. Our dissolved oxygen, fully saturated at the surface, and then right when we cross the interface, you end up sort of in hypoxic area. You get this little excursion where it increases up there, not really sure what's going on there. And then eventually you end up in fully anoxic conditions down at the bottom. pH as well. pH is lower, we're talking, well, I don't know what's going on there. Yeah, I don't know if this pH data has actually been QC'd yet. There might be an issue with that. But moral of the story is the pH water column and the sediments end up being very low in pH, and I'll show a little bit more of that later. Sulfide in iron data. So it's as easiest to sort of conceptualize if you start from the bottom. So this is hydrogen sulfide, this S2 minus. You can see it increases near the bottom of the blue hole just because we have a lot of sulfide, as you'll see in the sediments, and it's fluxing out. But then we get this beautiful, in my discipline, sediment diagenesis, this beautiful redox cascade. It's classic redox cascade, the same thing you would see in sediments. We're seeing it in the water column. So that hydrogen sulfide is oxidized to elemental sulfur, S0. And then right above that, that's being oxidized to thiosulfate right above that. At the same time, we also see iron chemistry as well. So there is some coupling between iron and sulfur systems. Of course, the iron is in pretty low concentration, as Tim could attest to. I guess it's more than trace, but still pretty low compared to the systems I usually work in. Interestingly, there was this big turbidity maximum down here, and what this ended up being is elemental sulfur. So we can actually remove a lot of elemental sulfur from this system by filtering. I think this is a post-filtering measurements, but we have evidence that that's basically what this turbidity maximum is. It's elemental sulfur. So I want you to, well, I'll talk more about that in a second. And lastly, our nutrients, again, starting from the bottom up, you got your ammonium coming up from the sediment pore waters, and that is oxidized to nitrate. And when I say, this is being oxidized to that and so forth, the same thing is in reverse, right? You could go from top to bottom and you could just have reductions of these various analytes. So there we have our two species of, and I say nitrate, but really this is nitrite plus nitrate. It's just the way we measure it. And finally, phosphate as well is enriched down here at the bottom. So when you look across the water column, you do see a gradient. So this right here is indicative of just even a diffusive flux, let alone any advection. So the blue holes would indeed serve as a source of nutrients, at least to the bottom waters around this area. This isn't a video, but we did in Amberjack find 2 dead sawfish, and FWC came out and scooped these puppies up. I don't know, is anybody, I was talking to a student who worked at FWC before. I wonder if it was part of the same,
Different arm.
Different arm, okay. But yeah, 2 dead sawfish, and how did they die? They suffocated underwater. It's kind of crazy to think about that, right? So they either didn't have their oxygen or they were poisoned by the sulfide, one of the two. And I don't know what this black stuff is. It's pretty gross. Oh yeah, and the FWC necropsy folks said it was the worst smell, like worse than any dolphin necropsy lab that they'd ever dealt with. So Nastasia Patton, our collaborator, she published this paper looking at the biological community, and sure enough, in the bottom water, this deep water, you do see that the microbial community does have some sulfate reducers. And we also saw a huge proportion of a previously undocumented Woes archaeota species. So you'll have to ask her if you want more details than that, because it's not my forte. Again, here is a sediment core, as I showed earlier. I know it's a busy slide, and I'll walk you through it. What you see at the sediment water interface is hydrogen sulfide accumulating in high concentrations, so millimolar concentration. So, you get these cores back and they reek. The whole lab is just, it just reeks. Low pH, we're going below 6. We see intense ammonium in DIC and DOC production. What's interesting is that phosphate, which you might be able to see in black here, is not produced proportionally. So, we would expect our phosphate profile to look like this, but instead we have phosphate removal. We suspect this is due to authigenic calcium phosphate precipitation, but not sure. Blue hole sediments, high in organic matter content, 5% or so on average, I think. And calcium carbonate, it forms the vast majority of the remainder of the mineral pool. I think that's all I wanted to mention on this slide. So to recap, in Amberjack, this interface where you go from sulfidic to anything else more oxidized. So sulfide, the sulfide redox interface is about 10 meters off of the bottom here. So keep that in mind, because that's going to be relevant when we talk about the next blue hole. Now, it was similar between the wet and dry seasons, so very stable. The redox chemistry, the microbiology, it was all pretty stable. And we didn't see any fluctuations in bottom water conditions over those 24 hours that the lander was deployed. Minimal fluctuation. So it doesn't seem like there's any evidence of flow. Nastasia contacted a physical oceanographer, David Constantinou, and he did some supposedly beautiful calculations, although I don't really have a context for what beautiful physical oceanographic calculations look like, but he said that it would require about 900 miles per hour for 36 hours to mix the blue hole. So, category 10 hurricane. Now, this was, really exciting for me, because we got a huge media storm that came about from this. It was, obviously a highlight of my career to have something like this, all this attention to a project. And blue holes actually got put on the map from the release of this PBS documentary. So it was really cool to see that happening. But now I'm going to talk about the green banana side, which, we didn't include in any of those. It wasn't in the documentary. It wasn't in any of those news articles. Now, you'll have to excuse the ugly Excel profiles here. I swear I had big aspirations to beautify this data set and present it for y'all, but life happens. So salinity-wise, I'm showing one deployment of the lander. In the lander, it goes down to the bottom, it sits there for 24 hours, and then it comes back up in the orange line. So salinity increases with depth. You do get some weird funkiness going on here on the way back up. Not sure why. Temperature, it gets colder down at the bottom. So you do get both salinity and temperature stratification contributing to that density stratification. O2, we don't get anoxic. We only get hypoxic. And we get higher FDOM in the blue hole. So fluorometric dissolved organic matter. Now this downcast was around noon and the upcast was around noon the following day. So keep that in mind, because as you're going to see, something happens during the night. And lastly, the electrochemistry results, which I don't have plotted because there's not too much to actually show for that. Unlike Amberjack Hole, we didn't detect hydrogen sulfide in the bottom waters. Nor, and we only detected trace levels of thiosulfate as well. So there is, some influence of sulfide, which is being oxidized to thiosulfate, but it was only in trace amounts, at least when we sampled at noon. Looking down at the sediments, we saw microbial mat at some sites. There was actually evidence too of what we think was a snail, although we can't tell from the grainy resolution, of the footage. So there is life down there. And so, you know, if this was a fully anoxic site, we wouldn't see life, presumably. Looking at our sulfide profile, it's not as clear cut as what we saw as Amberjack with respect to just being a constant increase. There's something going on here that's causing its removal. Again, pH drops, you know, to 5 or so, or in the 6s. The ammonium, I'm not showing the data for the other analytes. It's not processed yet, but ammonium displays this consistent increase with depth. And phosphate, interestingly, we actually see an increase in phosphate at the surface, but then a removal of phosphate at depth. So it seems like the sediments are a little more dynamic here at Green Banana, you know, potentially due to some input of aquifer water. There is evidence for flow just from the diver video, and the divers said that they saw flow or they felt flow. And I know the divers are moving, but you can still see it when they stop as well. So there's particles being moved around. All right, now y'all are gonna hate me for how busy this slide is, but I'm gonna walk you through it slow. I like seeing all the data at once when I'm looking at my data and, you know, haven't had a chance to make it in a user-friendly format. So what you're looking at here is a 24-hour time series. This is just a zoom of the top 3 plots on the left here. You're looking at this noon to noon time series of the bottom water conditions, you know. So what you see at exactly 8:36 at night is salinity, which is, oh, I got to catch my bearings, in green here. Your salinity, you drop about a half a unit. So you drop about a half a unit, and then you have these high frequency fluctuations in salinity until about 8 a.m. the following morning when it starts to rebound. What else do we got? We got temperature. Temperature increases slightly here in the cyan color. Again, you know, the high frequency fluctuations. We see 100-fold increases in chlorophyll and phycocyanin. Those are in cyan. Actually, temperature is this blue one. Cyan is the chlorophyll. So we see 100-fold increases in chlorophyll, phycocyanin, and FDOM, right? So whatever is presumably coming into this water is extremely dark and low salinity, and finally low oxygen. Our oxygen goes from about 15% saturation down to, you know, basically the detection limit of what the sensor is, just a couple degrees or a couple units of oxygen saturation percentage. Now, this is electrochemical data from those electrodes that we strapped to the both inside the benthic flux chamber as well as on the ambient water. Again, this ambient oxygen time series matches what we see up above, so that's kind of good news, right? Granted, the magnitude's a little bit off. I would trust the SON data over the electrochemistry for that. The chamber, the benthic chamber, of course, is a little encapsulated environment in contact with the sediments, so the chamber decreases soon, pretty quickly. You have a pretty rapid oxygen consumption by the sediments. Now this one, first things first, the ambient outside bottom waters, we have hydrogen sulfide here in green. So the sulfide, you know, you kind of see a few spikes here and there, but then all of a sudden, right when you get, you know, a little past that 8:36 p.m., you start seeing these, you know, again, high fluctuations from basically below detection limit up to a few micromolar. As time goes on, we see thiosulfate start to actually manifest. So presumably what you have is this hydrogen sulfide that's fluxing out, probably from the aquifer, and then that is reacting with some of this oxygen that's here in the ambient waters. And actually you do see a little bit of a decline in oxygen over time, suggesting it may be being consumed by the oxidation of this sulfide. And we see some of this thiosulfate being formed over time in blue in the bottom of the water, in the bottom waters. Now, I'm not going to go through the nutrient fluxes in detail, just because it's busy, takes a while to explain. But what we do see is intense nutrient fluxes in the benthic incubation chamber. So we see high organic nitrogen fluxes here plotted as STDN. You can do the math. We see high ammonium fluxes relative to the ambient background conditions. We see high DOC, well, DOC maybe not so much, but we see high phosphate relative to the external water time series. Now, I'm not showing this data from the dry season, but we don't see these, we don't really see any of these changes during the dry season. We don't see intense changes in any of these sort of more oceanographic water quality parameters. We don't see intense fluxes even in the benthic chamber. And so what we think, or what I think is going on, is you do have aquifer input, but it's percolating through the sediments and actually causing, you're increasing your flux into the benthic chamber just due to sort of this percolation. You're increasing advective input of these nutrients into the benthic chamber. You know, whether it's coming in laterally as well, it might be, but we don't really see substantial increases in nutrients, maybe like a little bit potentially for some of these, but it's not nearly as significant than what's in the chamber itself. So again, we see this in the wet season. We don't see this in the dry season. You know, we're not positive why this is happening, and it may be, we may not have enough data to actually be able to say, oh, well, it's a wet and a dry season, you know. Yeah, you don't see it in the dry season because there's no aquifer discharge or pressure or whatnot. The aquifer isn't loaded with water, but it could be related to lots of other things like phases of the moon, barometric pressure. So we can't necessarily rule out, you know, other controls than rather just wet or dry season. And when I say wet or dry season, it was September and May. So for the dry season, we waited all the way till the end of the dry season. Kind of same thing with the wet season. You know, we waited till the end of the season in order to give any terrestrial water time to kind of make its way offshore. So this is one of Chris Smith's ideas, and I may get this wrong, and I apologize, Chris, if you're listening. But what he thinks might be happening to control this. Oh, I forgot to mention a very important thing. So we saw that during the wet season, we saw that flux that happened at 8:36 p.m. at night. That was a Monday night. We deployed the lander on a Thursday night. 3 days later, we saw the same exact thing happen. It was almost identical. It just happened 1 minute later. So it is like you could set a clock by the blue hole discharge. So maybe when we're on shore, or maybe during the day, you know, you have an enhanced sea state, or you have a sea breeze that's pushing water towards shore, and you don't, you know, if anything, it's forcing water downwards into the aquifer. So it just resembles a more oxygenated environment like the water column. At night, you may get the opposite situation where you're actually almost pulling a vacuum and pulling this water out. You know, and it's not like it's purely fresh water. It was only a half a unit less in salinity than the other just water that was in the blue hole prior to seeing this apparent discharge. One important implication, if you're going out and looking at blue holes or doing any other sort of biogeochemical exploration, if you will, is, you know, if we were only out there during the day and we didn't have this sustained sensor package down there, we would have concluded that there was no flow. So it's pretty important to get those round-the-clock measurements. So I'll wrap it up. You know, again, this is a diagram that explains, again, that funnel shape, how you're capturing carbon when stuff dies and accumulates down here, and then it's remineralized, can flux upwards, cause primary production, and zooplankton, fish come, you know, life propagates laterally then from there. So maybe we need to make these things marine protected areas. You know, I mentioned the analogy for habitat restoration, terrestrial connectivity. We talked all about that. We did run a simple, just back of the envelope box model, and, you know, just the nutrient flux from this Amberjack, just from our diffusive gradient of nutrients, It could sustain a moderate bloom within a 1 kilometer squared area. So it does seem, oh, and Vince Loveco's group at USF did find not Karenia brevis, but Karenia mikimotoi. So there may be, you may need this cofactor species that may be required for Karenia brevis to take off. There's just a lot of circumstantial evidence, right? So certainly shouldn't rule out the red tide connection. The blue holes, they come in all different shapes and sizes, like Goldilocks. So there's this redox nutrient pH gradient both within the blue holes and then between the blue holes. It's useful for process paleo or future studies. And finally, make sure you measure 24 hours at a clip to catch these transient phenomena. So I'll shut her down. I'll take questions if we got time. I don't know.